After this lesson, you will be able to understand what is DNA fingerprinting, principle of DNA fingerprinting, process and various methods, and its applications. DNA fingerprinting is a laboratory technique developed by Alex Jeffreys, a British scientist. It is used to establish a link between biological evidence and a suspect in a criminal investigation. A DNA sample taken from a crime scene is compared with a DNA sample from a suspect. If the two DNA profiles match, then the evidence can be proved. Conversely, if the two DNA profiles do not match, then the evidence cannot be proved. DNA fingerprinting is also used to establish paternity disputes. The process was first used in forensic study in the solving of murder of two teenagers in 1983 and 1986. In India, DNA fingerprinting was started by Dr. V.K. Kashyap and Dr. Lalji Singh. Dr. Lalji Singh was an Indian scientist popularly known as father of Indian DNA fingerprinting. Although 99.9% of human DNA sequences are the same in every person, only 0.1% is different, and that is enough to distinguish one individual from another, unless they are monozygotic twins or identical twins. DNA fingerprinting uses locating the repetitive sequences that are highly variable called variable number tandem repeats or VNTRs, in particular short tandem repeats STRs, also known as microsatellites and mini satellites. VNTR loci are similar between closely related individuals but are so variable that unrelated individuals are unlikely to have the same VNTRs. A variable number tandem repeat or VNTR is a short nucleotide sequence organized as a tandem repeat. These can be found on many chromosomes and often show variation in length, number of repeats among individuals. Each variant acts as an inherited allele, allowing them to be used for a personal or parental identification. The process involves various steps. The first step is extraction or isolation of DNA. Before the DNA can be analyzed, it must be extracted from the cells and purified. A sample of blood, saliva, semen, vaginal lubrication, or other fluid or other tissue from personal use item can be used for extraction of DNA. There are many ways this can be accomplished, but all methods follow the same basic procedure. The cell and the nuclear membranes need to be broken up to allow the DNA to be free in solution. Once the DNA is free, it can be separated from all other cellular components. After the DNA has been separated in solution, the remaining cellular debris can then be removed from the solution and discarded, leaving only DNA. Second step, digestion of DNA by restriction endonucleases. The extracted DNA is large and need to be cutted into fragments with the help of restriction enzymes. These enzymes are also known as molecular scissors. Restriction enzymes cuts DNA at particular sequences called palindromic sequences. For example, an enzyme called EcoR1 will cut DNA only when the sequence that is 5' to 3' is GAATTC. Next step is separation of DNA fragments by gel electrophoresis. The DNA pieces are sorted according to size by a technique called gel electrophoresis. The DNA fragments are passed through agarose gel. The DNA has net negative charge due to phosphate group and moves toward the positive anode through the finer mesh of agarose under the influence of electric field. The smaller the fragment moves the fastest in the gel and separated based upon the size. Fourth step is Southern blotting. Transfer of electrophoresis separated DNA fragment to a filter membrane to nylon or nitrocellulose membrane is known as Southern blotting, named after the scientist E. M. Southern. The distribution of DNA pieces is transferred to a nylon sheet by placing the sheet on the gel and soaking them overnight. Next and most important step is hybridization using VNTR's probe. Radioactive or colored probes are added to nylon sheet, produce a pattern called the DNA fingerprints. Each probe typically sticks in only one or two specific places on the nylon sheet. Membrane is washed repeatedly to remove non-specific binding and to clear the background. Last step is detection of hybridized DNA fragments by autoradiography. The hybridized DNA fragments with radioactive probes forms a pattern detected by autoradiography. These DNA probes makes pattern like barcode called DNA fingerprints. These patterns are compared with suspect DNA that also undergo the same process, and then they are analyzed and interpret with those of the suspect. Application of DNA fingerprinting. First is biological evidence to identify criminal. Where fingerprints are not available, but biological specimens are available like blood or semen stains, hairs, or items of clothing at the scene of the crime, then these items may prove to be valuable source of DNA of the criminal. Since the year 1987, innumerable cases have been solved with the help of DNA fingerprint evidence. Second is paternity disputes. Another important use of DNA fingerprints in the court system is to establish paternity in custody and child support litigation. In these applications, DNA fingerprints bring nearly perfect evidence in paternity disputes. Third is personal identification. DNA may be the best way to identify a person as all the body tissues and organs contain the same DNA type. The specimen required also is very small. In fact, the US Army has been doing DNA fingerprinting of all its soldiers and has a huge data bank. Now time to check your knowledge. Who developed the technique of DNA fingerprinting? Yes, correct. It is Alex Jeffreys, a British scientist. What can be the possible source of DNA for DNA fingerprinting? Yes, correct. It is blood or semen stain or hair or items of clothing at the crime scene. Next question is transfer of DNA fragments from agarose gel to nylon membrane is known as northern blotting, southern blotting, western blotting, or hybridization. Yes, correct answer is southern blotting. While the term northern blotting is used for RNA and western blotting is used for protein. So correct answer is southern blotting. To locate the VNTRs in DNA fingerprinting, small oligonucleotides are used that can be radioactive or colored, known as, yes correct, these are known as probes. At last, we recall what we learned from this chapter. First, DNA fingerprinting was developed by Alex Jeffreys. It is based upon detection of VNTRs that show polymorphism. DNA is extracted from the sample. Then this DNA is digested using restriction endonucleases. Once these DNA fragments are digested, they are separated using agarose gel electrophoresis. The separated DNA fragments are transferred to nylon membrane, a technique called southern blotting. Then they are hybridized using small oligonucleotide that can be radioactive or colored, called probes. And finally, they are analyzed using autoradiography and compared with the suspect.